So today I'm going to be talking about subcortical brainstem systems that are involved in regulating sympathetic nervous system activity. And this is part of work that was led by Tor Wager. I'm currently one of his postdocs. And it's really focused at building an online wiki or an interactive resource so different researchers that may not have core expertise in neuroimaging can leverage this information to facilitate sort of transdisciplinary research and investigating these brain systems from diverse points of view. So how does the brain mediate changes in the peripheral nervous system, in particular sympathetic nervous system activity? So as you can see here on the left of the screen, this is a complicated process, but you can break it down into 4 basic components, where you've got higher order cortical systems evaluating threat and sort of the body's ability to cope with this threat in the environment, and then based on that information, you could then have processing that goes down to the viscera and target organs through visceromotor channels through the autonomic nervous system. And this is mediated in part by the brainstem. So there are 2 important components of this model, but moving beyond that, sort of the factor that might be more important in terms of cancer research is how chronic changes in target organs may be influenced by these visceromotor channels, where if you have sort of chronic stress evaluations, you have higher pathogenic risks or maybe potentially negative outcomes in terms of disease progression. So in terms of this model, what's the current evidence in terms of the neuroimaging literature on how these central higher order and brainstem brain regions are associated with changes in sympathetic or parasympathetic nervous system activity? So on the right, just plotting a simple review of the literature. This is about a year ago, showing where just peak locations from neuroimaging analyses, where they're either positive associations with sort of peripheral measures that are associated with increased sympathetic activity, so that's general cardiac activity increases or heart rate variability increases, and sort of the lower frequency spectra of heart rate variability, and then alternatively brain regions that are associated with parasympathetic increases, and that's going to be reflected in decreases in heart rate generally or changes in high frequency heart rate variability. So as you can see, there's no very clear mapping here, but sort of at a broad scale, you can see that more dorsal regions of the prefrontal cortex are associated with increased sympathetic activity, and more sort of subgenual and ventromedial prefrontal regions are associated with parasympathetic modulation. In terms of brainstem structures, there's not so much of a tight-knit picture as well. There's still some general associations in periaqueductal gray, amygdala, and pons with increased sympathetic activity. But in general, neuroimaging using conventional methods, it's really hard to tell what specific nuclei are driving these changes. And so it's hard to get a clear picture of the exact pathways from higher order cortical regions to peripheral sort of measures that we might be interested in measuring. And so one of the main reasons why that's an issue is because conventional standard like 3 Tesla magnetic resonance imaging doesn't get you the spatial precision that you need to really look at these brainstem structures. So because in general, many of the brainstem nuclei are small on the order of 1 millimeter in scale, and the point spread function of standard bold at 3 Tesla is around 4 millimeters, you can't really just be sure that you're measuring these nuclei with the precision enough to be confident that you can assign this functional localization to the region you might be thinking you're measuring. So on the right here, I'm actually plotting some results from a study published in Science where there's a bit of a debate regarding studies that thought they were imaging the locus coeruleus in the brainstem, and that, there are these clusters of activity and sort of the posterior brainstem regions where the authors thought they might be correctly identifying locus coeruleus, but it's inconsistent with neuromelanin imaging, which is just based on the amount of neuromelanin in the brainstem, and you can pick that up with structural MRI. And these 2 different modalities, while they should co-localize in the same region, they give sort of this impression that functional imaging of the brainstem is not as precise as it needs to be to really get it at this sort of strong claims about what systems are being engaged. So these sort of problems have caused recent researchers to use more ultra-high field MRI. So this is MR at 7 Tesla or higher to try to get better localization of these brainstem structures. Here I'm just showing a number of regions from Biancardi et al. Where they used different functional approaches, so using bold imaging with T2 star and functional or fractional anisotropy to look at a functional atlas based on functional MRI time series, which could better differentiate these sorts of brainstem structures. In addition to sort of localizing these different types of structures, studies have looked to link brainstem activity to actual peripheral measures that are concurrently measured during fMRI. So this is just one example from the literature that's looking at responses to painful stimulation using a pressure cuff. And additionally, looking at cardiorespiratory measures to get at heart rate variability. And so they found, consistent with the classic brainstem models and pathways in animal models, that activity in rostroventral medulla actually tracks the amount of high-frequency heart rate variability measured and decomposed from cardiorespiratory data. And so that's really consistent with these animal models, and it's sort of a piece of evidence that advancing these neuroimaging methods can sort of leverage information and sort of build evidence towards confirming these pathways. Another study using not high field MRI, but still using focused field of view to get at brainstem imaging. So this is at 3 Tesla with slightly larger voxels, showed that spontaneous muscle sympathetic nerve activity during MRI was correlated with spontaneous activity in rostroventrolateral medulla, and also with decreases in activity in caudal ventrolateral medulla and the NTS. So these brain regions are classically known as part of the baroreceptor reflex, which is involved in regulating blood pressure. So if your blood pressure decreases over time, you'll see the same pattern of response to increase cardiac output and to regulate your homeostatic needs. So these two studies together really show that functional imaging can be used to sort of track these processes, and that it's important to integrate knowledge across these diverse types of studies to really get at what's reliable and sort of in terms of brainstem imaging, as opposed to what may be sort of up in the air. So part of the lab's work really here is to develop this wiki to sort of provide an interface for this knowledge, where we can look at cascading from sort of higher cognitive brain regions from frontal cortex to basal forebrain down to the brainstem at the macro scale, and provide an interactive sort of guide to these brainstem pathways, where interested individuals can go and highlight any particular region and get information that is either driven from the literature or based on literature review or in a data-driven way. So moving on to the specific key features of this wiki, I'm going to use the periaqueductal grade as an example and highlight the main points and sort of information that you can get from this website. So the key features include information about localization, so both in terms of just providing anatomical illustrations of all the regions. Additionally, reviews that show functional activation, what it might look like, and whether or not imaging data you get would be reasonable or not. Additionally, in terms of localization, we provide regions of interest for neuroimaging analysis, and additionally, study-specific coordinates, so you can pinpoint specific regions that were found depending on the context of individual studies. Additionally, we've automated capturing data from the field to get sort of lists of references depending on key coordinates from this NeuroSense framework, and additionally have a hand-curated set of important studies for each of these brain regions that you can download, and these are available just in EndNote format. In addition, there's information in terms of these pathways, both in terms of afferent and efferent connections, in terms of major reviews in the literature, and beyond that, just providing information about the major functions across diverse disciplines. So, in summary, this sort of work fits in a framework that shows the importance of brain-body pathways in terms of physical health and can hopefully connect researchers that are interested in understanding how different parts of the brainstem interface between higher cognitive functioning and peripheral outcomes. Thanks. And I'd like to thank my lab and the contributors who put a lot of work into developing the content for the wiki, and Torwager and the funding for the project. So, thanks.